Today I'll be discussing about my study titled LocusMasterTE Integrating Long-Read RNA Sequencing Improves Locus-Specific Quantification of Transposable Element Expression. Transposable elements, which are genomic segments that are repetitive in the human genome, exist in many copies. They have ability to move and jump around the genome. Based on their movement mechanism, transposable element can be divided into two different classes. The class 1 element uses a copy and paste mechanism, and class 2 element uses a cut and paste mechanism. Inside class 1 elements, T's can be further divided into LTR, sign, line, and SVA based on their sequence composition. And looking at this pie chart here, TEs actually make up half of the human genome. Lines are the most frequently prevalent, followed by the sign and then LTR, and then the other parts are like DNA transposon or SVA, etc. So even though, as I mentioned, that transposable element Ts are capable of jumping around the genome, many copies have lost their ability and now remain at fixed genomic position. And researchers have identified these fixed positions and compiled them into a really nice comprehensive database called Repeat Masker Database. To briefly introduce about this database has quite a lot of information, but I'd like to highlight two different information that the Repeat Masker has. The one is about the T classification. As I mentioned, TEs have a lot of copies in the human genome. So they classify further TE locus into class, family to subfamily based on the sequence composition. So if you look at the third row, the TEs are located in the chromosome 1 from here to here. And then the subfamily that it belongs to is L1MC5A. And this is also one subfamily belongs to line class. And as I mentioned, the grouping is all based on the sequence similarity. So within the same subfamily, there are high consensus, like there's high similarity and also consensus sequence exists. Second important piece of information is that the estimated age of the Ts, which is determined by measuring how much their sequence have diverged from the original copies, which is in this millidiv column. So lower the number is, it means that the low divergence value indicates younger elements. And age is very important because newly inserted elements or younger TEs are generally considered to be retain more activities compared to the other older copy. Although like TEs are composed of approximately 50% of the human genome, many of them are actually silenced in the normal setting with the various level. It can be silenced by DNA methylation. Also, after DNA methylation, there's another level, which is at the RNA interference level using like siRNA or siRNA or piRNA. And there's a histone modification, which will regulate the TE expression. However, because in the cancer setting, there are dysregulation happening. So therefore, these T that were originally be silenced are become reactivated. And sometimes after the reactivation, T will produce a TE containing mRNA like this, and from 1 to 3. And sometimes even line elements can cause for a protein, and this will lead to a double-straight DNA breakage. Or another one kind of the TE, ALU, which is one kind of design elements, can be formed a double-strand RNA, and this will lead to A2I RNA editing as well. So acknowledging like this kind of all different importance of the TE, we then looked at how the TE, what's the meaning of having a product from the TE? So those products that has been generated by the reactivated TE can be further recognized by the innate immune responses, and we call this as a viral mimicry. Sometimes some of the TE can even produce like a peptide or envelope protein, which can be further recognized by the T cell or B cell. So with this importance of the TEs in cancer, we aim to quantify TE in cancer sample. However, one of the major challenges of studying is about their repetitive nature. Because TEs are the genomic unit that has a very repetitive segment, which will create the significant difficulties especially when you use a short read RNA sequencing data, because the technical read lengths are short, which means many reads happens to map to a multiple location across the human genome, and these are referred as multi-map reads. To address this issue, conventional tools have employed an expectation maximization model, EM model. An EM model is an iterated optimization technique that used to estimate the parameter of the probabilistic model when the data contain hidden variable or is incomplete. So particularly useful in a case like the one that I've showed you, like the multi-map cases, where you can directly compute the maximum likelihood estimate, MLA, to trying to identify the missing information in it. So by using this EM model, researcher can better allocate the multi-map reads to their most likely genomic origin, improving the accuracy of T quantification. However, we also thought of that maybe with the development of the new technology, we believe that long read has a better opportunity than the short reads, because long read has a longer technical length, offering a significant advantage in capturing T expression. One of the major advantages is that it will have a chance to reduce the issue of the multi-map reads, which is a very common challenge in the short read RNA sequencing. So to briefly introduce about the general background of the long read RNA sequencing, for not familiar with it, there are two major sequencing companies that are actually leading this technology, PacBio and Oxford Nanopore. Each utilize a very distinctive method from their own for generating a long read RNA sequencing. The long read RNA sequencing can produce reads up to 2 megabytes at one run read, although the average read length is typically ranging from 1 to 100 kilobases. This range is particularly well suited for capturing like full-length transcript or of a splice gene, which is usually like 1 to 3 kilobases. So given these properties, our hypothesis is that longer technical read lengths of long read RNA sequencing may provide a better solution to overcome the multi-mapping issue associated with the short read RNA-seq, so which will thereby improving the accuracy of T quantification. To further confirm our hypothesis, whether long read is actually having less multi-map reads, we use our in-house generated HCT116, which is a colon cancer cell line, and run for both short read and long read. In this case, the long read is a nanopore, using a nanopore technique. So our results shows that long read has significantly lower multi-map proportion than the short read does. And we also looked at how much the correlation is happening between the long read and short read captures at TE subfamily and TE individual level. In the TE subfamily level, we do see there's a good correlation between the long read and short read captures TE quantification, but in terms of the TE individual, the correlation is pretty bad. So we think, and we observe high discrepancy between short read and long read T quantification. And moreover, one thing we have identified is that we identified an existing limitation in the current EM model that conventional tool has. In theory, EM model should converge after a certain number of iterations and assign each one read to a single genomic position. However, we found there's a 6% of the cases of the EM model fail to resolve the multi-mapping issue. While the 6% seems to be very negligible at first glance, when we look at the age of the 6% of those unresolved T, we discovered that those T's are significantly younger than the T's for which the EM model successfully reached to a conclusion. This result is actually aligned with our expectation, because younger TEs will have not accumulated enough unique sequence over time, making it more challenging for EM model to distinguish them from other copies of the human genome. And in contrast, if you think about like an older TE, Typically they will have much more unique sequence after as the time accumulated, which makes EM model to be easily resolve a multi-mapping issue. Additionally, we compare the ability of long-read RNA sequencing and short-read RNA sequencing to capture younger TEs. Our analysis confirmed that long-read is indeed better at detecting the younger TEs, likely due to their longer read lengths, which reduce the ambiguity associated with the multi-mapping. So to address the challenge in the T quantification, we have developed a tool called LocusMasterTE. Essentially, what our tool does is it integrates long-read information as a prior term into the EM model while quantifying for the short-read RNA sequencing. The reason why we still want to quantify TE at the short-read is that many of the cancer core, like CPTAC or TCJ, are in the short read. So if we want to find some of the findings from the clinical or like other epigenetic markers or like in the big cohort, we definitely need to develop a tool that will accurately quantify T in the short read RNA sequencing. So here's a schematic overview of the LocusMasterTE pipeline, illustrated step by step. So figure A. First, short-read RNA sequencing is aligned using a provided annotation file. And based on the alignment score, the reads are classified into uniquely mapped reads and then multi-map reads. And it will assign differently in the individual T copies. In this case, it's T, A, B, C. Next, the long-read RNA sequencing. Any of the PacBio ONT actually works. Is aligned with their own alignment tool. And then the quantification will be happening using a feature count utilizing a same annotation file you use it for the short-read RNA sequencing. And then initial proportion will be calculated for each of the T copies. And then this information calculated from the long read will be normalized and it will convert into a transcript per million, and this will be integrated while the multi-mapping will go through the expectation maximization model. And then after the model will converge, it will reassign the reads based on the conclusion given by the model. So one of the things that I want to highlight is that because our tool is not, except for the integrating a parts of the long read parts, other parts are very conventional approach that many T quantification tools already have. So what we essentially did is that we take one of the widely used conventional tool called telescope, and then we develop a LocusMasterTE upon the telescope, basically having additional parameters which will be able to integrate the long read during the EM model. So essentially, most of the analysis from now on will be comparing between the telescope and then the LocusMasterTE. So briefly to just go from the evaluation of the precision and recall of how LocusMasterTE will quantify the TE. We evaluate the accuracy of our tool. We generate a simulated short-read RNA sequencing from a long-read RNA sequencing derived from the HCD116 cell line. We then compare the performance of the LocusMasterTE and other several conventional methods. And we do see that LocusMasterTE has achieved highest precision and recall over other conventional tools in quantifying transposable elements. And another thing that we also examine is whether LocusMasterTE reduces the instances where the EM model fails to resolve the single origin of the read, which means effectively addressing a multi-mapping issue. So with using a LocusMasterTE, we identify there's the unresolved cases were reduced down to 1%, which is a significant improvement. Additionally, we introduced a parameter called long read weights, which allow user to manually adjust the contribution of long read information in the model. These features are very useful because we also trying people to use our tool whenever they have like a long read RNA sequencing coming from a different tissue from the short read as well. So by adjusting this parameter, user can manage the influence of the long read information while quantifying for the short read RNA sequencing. So lower weights are recommended when the long read RNA sequencing will be originate from a distant tissue type from the short reads, but the optimal weight is around one, um, uh, around one. And to test this, we conducted a several simulation using a various long read weight value ranging from 0.01 to 1.5. And for this test, we utilize a in-house ACT116 short read RNA sequencing along with the K562 long read RNA sequencing from a SGNX study. So we do see that whenever you use it with the distant tissue type, it's recommended to use a value smaller than 1 for the weight parameters. And also, finally, we trying to ensure the compatibility between the two major long read RNA sequencing technique, nanopore and PacBio. And we compare the performance of LocusMasterTE using a short read simulated, previously mentioned simulated short read RNA sequencing. And then we compare the TE quantification that has been generated by the LocusMasterTE with the two different input for the long read RNA sequencing. And we do see that there's a quite good correlation, although there's a slightly variation in here, but in general, there is a quite good correlation with 0.9 Spearman rank correlation coefficients. And this indicates the both ONT, nanopore, and also PacBio serve as a suitable long read inputs for the LocusMasterTE. So to demonstrate the advantage of using the LocusMasterTE for setting T quantification, we sort of want to identify what sort of a feature that are significantly captured by the LocusMasterTE. So we analyze 6 different cell line samples. We have a one ACT116 from our in-house, and we have like 5 different cell lines from the SGNEX dataset. So essentially, we have 2 ACT116 and 1 A549, 1 HAPG2, and then 1 K562, and 1 MCF7. So we focus on TE that shows expressions between the expression differences between LocusMasterTE and Telescope. And interestingly, these T's are generally younger, like the TEs that were corrected by the LocusMasterTE, meaning the T's will have a T will have a different counts between the LocusMasterTE, and Telescope is much younger than the ones that does not have any changes in between the LocusMasterTE and Telescope. And this finding suggests that LocusMasterTE is better at correcting the expression of younger T, which are typically more challenging to quantify. So to further validate our tool, we compare the T counts generated by the Telescope and LocusMasterTE with result from the long read RNA sequencing. So this is the in-house ACT116. We observe an increased correlation in the T expression with the long read result when using a LocusMasterTE compared to the Telescope. And this is further confirmed by the other 5 cell line samples from the SGNEX dataset. And this confirms that there's a successful integration of the long read RNA information in LocusMasterTE, which will improve the accuracy of the T quantification in cell line samples. And another thing that we also wanted to check is the histone modification. So because histone modification is one of the mechanisms that will regulate the TE, and they will have a really whether it's marker for the active or repressive, it will have a strong correlation with the TE activity. So we try to explore this. We selected the TE and we grouped them into 2 based on which methods reported higher expression value for each. So brown represents TEs, TEs that has a higher count in the LocusMasterTE, and the blue represent TE that has a higher count in the telescope. We then analyze the association of these TE2 groups with 5 different histone markers, 3 active markers, and then 2 repressive markers. So each A3K4ME3, A3K9 accelleration, and A3K20 acceleration are the 3 major active markers. So, and then just to make sure there is no potential bias in histone data, we restricted our analysis to T region with the mappability score greater than 3.5. And we compared the highly captured by the LocusMasterTE shows less enrichment for 2 of the repressive markers, and greater enrichment for all 3 active markers. And but this is just verified in the ACT116, and we also have a supplementary figure which will see a very consistent trend for the other 5 cell line samples from the SGNX dataset. So this also suggests that LocusMasterTE improves T quantification by providing results that are more closely aligned with the independent epigenetic data, especially the histone markers. Okay, the final thing that we did is that we tried to validate the reads reassigned by the TEs by LocusMasterTEs are generally from the TE derived. So what we did is that we analyzed the RNA editing events in these samples from the TCGA colorectal cohort. RNA editing is a post-transcriptional modification of RNA sequence mediated by specific enzyme. So notably, like the recent study have shown that allele elements, which is one type of the TEs, accounts for over 99% of the human RNA editing events as they form a double-strand RNA structure, which will undergo the A2I editing. So we use the TCGA colon cancer data, and we looked at, we also used the reference. Reference is generated, reference meaning the RNA editing trinucleotide signature derived from the MDA5 protected allele elements and 5 AZA treated AZA wild type samples. So which will basically showing the trinucleotide essentially originated from the allele elements. And then we compare with the 3 different groups, starting from a coding genes as a negative control, and then we group the TEs into 2. The common TE means TE that are both captured by the LocusMasterTE and telescope. And then we also have a group that are essentially only captured by the LocusMasterTE. So between the common and then reference and the LocusMasterTE, we see there's a very highly similar things happening with the reference, displaying a characteristic peaks as CTA to CCA, so or CTG to CCG, or GTG to GCG. And this similarity was further supported by the high cosine similarity in here. However, in the contrast of the coding gene, which will serve as a negative control, has a very distinctive patterns in the trinucleotides and also lower cosine similarity. All these findings suggest that the reads reassigned by the LocusMasterTEs are indeed derived from the T-associated RNA that will undergo the RNA editing and further validate the that LocusMaster is actually capturing the expression that are generally from the TE expression. So to conclude my talk today, we developed a LocusMasterTE which leveraged long-read RNA sequencing as a prior term in the EM model to resolve multi-map reads in the short-read RNA sequencing dataset. And LocusMasterTE can be supported by both PacBio and Nanopore sequencing. And we can also tune user can also tune the weight for the long-read RNA sequencing so that they can utilize any available long-read RNA sequencing as a long-read input. And what we have observed is that LocusMasterTE is better at capturing the younger elements, and those activities also corroborated by the histone markers. And the detected Ts by the LocusMasterTEs has a very good, very solid RNA editing signature valid in the TCGA colorectal colon cancer. So I'd like to thank my lab members, and these are the people, especially the people that will help me throughout my research, and especially I would like to give a biggest uh, gratitude to my supervisor, Professor Jason Wong, who is always giving me a insightful advices throughout the research. Thank you.